Now we will talk about the sexual and asexual propagation in plants. In sexual reproduction, male and female cells or gametes unite to form a single cell, called a zygote. This zygote then undergoes cell division, ultimately giving rise to a new plant body. So shown in the illustration is a simple flower with different parts. Its parts include stamen, carpel, sepal, receptacle, and then petal. These are the parts of the flower. So we have the stamen, which function as the male reproductive part of the flower. Then we have the stigma, sticky surface to the ovary through which pollen tube grows. Then we have the style. It links the stigma to the ovary through which pollen tubes grow. Then we have the ovary contains ovules which develop into seeds and fertilized. And then you have the receptacle which enlarge end flower stalk on which other parts of flower are being born. Then we have the pedicel, the stalk of the flower. And then we have the sepal which protects the flower while in bud. Then we have the petal, often large and colored to attract insects and many more parts. Seen in the illustration is the role of ovary and ovule in reproduction. So first, there is the stigma, which pollen grain enters and then which then extends a growing pollen tube down the style into the ovary. Sperm cells produced in the pollen grain then move down the tube and make contact with one of the ovules in the ovary, ultimately reaching the egg inside. So this fertilization results in the production of a seed which can grow into another. Now we'll talk about pollination. It is a process by which pollens are transferred from anthers to the stigma of a plant or flower, thereby enabling reproduction and fertilization. So pollination has two types. We have here the biotic pollination and the abiotic pollination. And then it has two processes, namely self-pollination and cross-pollination. Biotic pollination is the process of pollination which requires pollinators, organisms that carry or offer the pollen grains from anther to the stigma. So, biotic pollination is done by insects mostly, but also vertebrates like bats, birds, and us humans. And we have also abiotic pollination, which refers to the situation where pollination is mediated without involvement of other organisms. So it occurs by wind and sometimes by water in case of aquatic. Self-pollination is the process of transferring pollen from the anther to the stigma of the same flower. It is the primary type of pollination in plants. Self-pollination is common in plants that contain male and female sexual parts in the flower. So, beans, peas, lettuce, tomatoes, peppers, eggplants are examples of self-pollinating plants. So, shown in the illustration, the letter A, it is how self-pollination happens. Cross-pollination is the transfer of pollen grain from the anther of one flower to the stigma of another flower. This type of pollination is complex, involving flowers that are genetically different from each other. So shown in letter B is the cross-pollination. Now let's move on to asexual reproduction. Asexual reproduction occurs when an organism makes more of itself without exchanging genetic information with another organism through sex. These are the types of asexual reproduction. So we have here the fragmentation, the spore formation, the vegetative propagation, the layering, the cuttings, and then the grafting. Fragmentation is a type of cloning where one organism is divided into minor fragments. Once divided, these fragments develop into individual ones which are fully grown. So shown in the illustration on how to do a fragmentation. So first is by cutting it from the mother plant, and then the cutted part, which is the shoot, will be placed in a water, I mean in a container with water, and then after how many days or weeks it will produce roots. Spore formation is an asexual reproduction technique where the plant produces hundreds of spores and the spore sacs burst, so the spores are dispersed into the air. So shown in the illustration is an example of spore formation in rhizopus. So we have here the hyphae and sporangium where the spores are located, and when it bursts, the spores will disperse into the air. Vegetative propagation. So it is the process in which new plants are grown from the old parts of another plant like roots, shoots, and leaves without involving any reproductive organ. So shown in the illustration on how to do a vegetative propagation. So first is cutting a shoot from the mother plant and then placing it in a pot with soil. So after how many days or weeks it will produce into a new plant. Layering is the process of bending a low-growing flexible stem to the ground. So it is done by bending a stem to the ground and then by covering it with soil so that it will produce roots. Grafting is a technique practiced by cutting, joining the parts of the two different plants and rooting in the ground. After some days, the tissues of the graft become integrated with the tissues of the rooted plant and develop as a single plant over time. So shown in the illustration on how to do the grafting. So first is choosing a shoot from another species of plant and then transferring it to another species of plant and then tying it. Now we'll talk about plant embryology. Plant embryology cover the anther and pollen development. Anther and pollen development have been widely studied as it is a complex and important process that leads to the release of viable pollen and plant fertilization. So shown in the diagram is the process of anther and pollen development. So we have here the microsporocyte stage, where diploid cells in the anther wall differentiate into microspores mother cells. The microspores mother cells are large round cells with a prominent nucleus. Then next we have the meiosis. Microspores mother cells undergo meiosis, a two-division reductional process that results in four haploid microspores. So we have the meiosis one. The homologous chromosomes pair up and exchange genetic material through crossing over. So the paired chromosomes separate and move to opposite poles of the cell. Then we have the meiosis two. The sister chromatids separate and move to opposite poles of the cell. This process results in four genetically distinct microspores arranged in tetrad. In the tetrad stage, the microspores remain encased within the tetrad for a short period, and then the callus wall of the tetrad begins to break down. Then we have the free microspore stage, where the microspores are released from the tetrad and begin to develop individually. Each microspore has a single nucleus surrounded by a thin wall. Then we have the vacuolated microspore stage. A large vacuole forms within the microspore, pushing the nucleus one side to one side. This vacuole serves as a storage organ for nutrients and water. Then we have the pollen stage. The microspore nucleus undergoes mitosis, forming a larger vegetative cell and a smaller generative cell. The vegetative cell is responsible for the growth and development of the pollen grain, while the generative cell is responsible for producing the sperm cells. Next, we have lastly the mature pollen stage, where the vegetative cell expands, filling most of the pollen grain with cytoplasm and organelles. The generative cell divides mitotically to form two sperm cells, and then the pollen wall thickens and becomes resistant to desiccation, preparing the pollen for dispersal. Covered also by the plant embryology is the ovule development. The ovule is part of the makeup of the female reproductive organ in seed plants. It's the place where female reproductive cells are made and contained, and it is what eventually develops into seed after fertilization, only for the seed to ripen and produce a complete adult plant. Shown in the illustration is the ovule primordium. So the ovule begins as a small bump on the inner surface of the carpel. We have the integumation initiation, where the two integuments grow around the ovule primordium, enclosing it except for a small opening called the micropyle. Differentiation of nucellus and chalaza. The inner part of the ovule differentiates into the nucellus and the base forms the chalaza, where the vascular bundle enters. We have here the megasporogenesis and megagametogenesis. A single megaspore mother cell undergoes meiosis, producing four megaspores. Three of these megaspores degenerate. Only one megaspore survives and undergoes mitosis, forming the female gametophyte or the embryo sac. The remaining megaspore undergoes mitosis several times, forming the female gametophyte, also known as the embryo sac. Then the embryo sac contains seven cells: the egg cell, the two synergids, two polar nuclei, and the three antipodal cells. This is now the mature ovule. The fully developed ovule comprises the following distinct parts. So are shown in the illustration. So we have the integuments, which protect the developing embryo. Then the nucellus, which provides nutrients to the embryo sac. And then the chalaza, which supplies water and nutrients. And then the embryo sac itself, which houses the female gametophyte, including the egg cell.
So moving on for plant embryology, the third one is seed development. I have listed here 6 stages for seed development. The first stage is double fertilization, followed by embryo development, endosperm development, seed maturation and dormancy, germination, and lastly, stage 6, which is seedling development. So for stage 1, which is double fertilization, double fertilization is a unique process found in flowering plants that plays a vital role in seed development. It involves the simultaneous fertilization of two distinct cells within the ovule, leading to the formation of both the embryo and endosperm. For first fertilization, one sperm nucleus fuses with the egg nucleus, forming a diploid zygote, which develops into the embryo of the seed. For second fertilization, the other sperm nucleus fuses with two polar nuclei in the central cell of the ovule, forming a triploid endosperm nucleus. So this ultimately develops into the endosperm, a nutrient-rich tissue that nourishes the developing embryo. So for stage 2 embryo development, The zygote undergoes a series of cell divisions and differentiation, giving birth to the root, shoot, and cotyledons. This structure becomes the foundation of the future plant. So the root, as we all know, is the anchor that absorbs water and nutrients from the soil. The shoot is the stem that gives rise to leaves, flowers, and fruits. Then cotyledons, the embryonic leaves that store food reserves for the developing seedling. For stage 3, we have endosperm development. So alongside the embryo, a triploid tissue called the endosperm develops. This tissue stores vital nutrients like carbohydrates, proteins, and oils, which provide essential nourishment for the growing embryo. The extent of endosperm development varies across plant species. In some, it's completely absorbed during development, while in others, it persists as a storage source within the mature seed. Stage 4. Seed maturation and dormancy. So, as the seed matures, its coat hardens, forming a protective barrier against harsh environmental conditions like desiccation, mechanical damage, and pathogens. So the embryo enters a stage of dormancy where its growth is suspended. So this dormancy ensures that germination occurs only in favorable conditions, maximizing the chances of seedling survival. So let's proceed first on stage 5, which is germination. So when exposed to the right combination of moisture, warmth, and light, the seed awakens from its slumber. The seed coat softens, allowing the embryo to break free and emerge as a seedling. So the cotyledons provide initial nourishment until the seedling develops its own leaves and begins photosynthesis. So the last stage, which is seedling development, so the young seedling continues to grow and mature, eventually transforming into a fully functional plant, capable of reproduction and perpetuating cycle of life. So the process of germination and seedling development is a marvel of nature, showcasing the remarkable resilience and potential for life hidden within a tiny seed. So by understanding this process, we can improve agricultural practices, conserve endangered plant species, and appreciate the wonders of, the natural world. So it's important to note that specific details of germination and seedling development can vary significantly between different plant species. Some seeds require specific treatments to break dormancy, while others germinate readily under optimal conditions. So moving on for plant embryology, we have embryology and systematics. So as we all know, embryology is the study of plant embryo development, and systematics is the science of classifying organisms based on their evolutionary relationships. So these two are intertwined fields that offer valuable insights into the fascinating world of plant evolution and diversity. So a closer look at their relationship. Embryology as a tool for systematic studies. I have listed here 4: homologous structures, synapomorphism, heteroblasty, and comparative embryology. So the first one is homologous structures. Embryonic features shared by different plant groups can indicate shared ancestry. For example, the presence of 2 cotyledons in most flowering plants suggests a close evolutionary relationship between these groups. The second one, synapomorphism, shared derived characteristics that arose in a common ancestor and are absent in more primitive groups. These shared features provide a strong evidence for evolutionary relationships. The third one, which is heteroblasty, the presence of different embryonic stages within a single plant species can shed light on evolutionary transitions and adaptations to diverse environment. Then lastly, comparative embryology. By comparing the embryonic development of different plant groups, researchers can identify evolutionary trends and reconstruct phylogenetic relationships. So, additional lang, examples of embryological data used in systematics, number first number and position of cotyledons, endosperm development, embryo morphology, and developmental rates and patterns. So the last part of plant embryology is embryological techniques. So embryological techniques serve a variety of purposes ranging from fundamental research to practical applications. So the examples that we have listed are microscopy, histological techniques, biochemical techniques, cytological techniques, in vitro culture, and genetic analysis. So for microscopy, we have here light microscopy, which provides a basic overview of embryo morphology and cellular organization. Next is electron microscopy and confocal microscopy. For histological techniques, we have here tissue fixation and embedding, sectioning, and staining. For tissue fixation and embedding, we preserve the embryo structure for detailed analysis. The third example, which is the biochemical techniques, we have here protein extraction and analysis, enzyme assays, and gene expression analysis. The fourth one, which is the cytological techniques, we have here cell isolation, chromosome analysis, and in situ hybridization. The fifth one, in vitro culture techniques. For embryo culture, it allows the manipulation of embryo's environment to study its developmental response to various stimuli. Then lastly, the genetic analysis. We have here gene knockout and overexpression, which manipulates gene expression to study the function of specific genes in development, revealing their roles and potential applications.
So hello everyone. So let's talk about the pollinology. So this is kind of new to me, so let's learn about this. So the pollinology is the study of the plant pollen, spores, and certain microscopic plankton organisms, collectively termed as pollinomorphs, so in both living and fossil form. So I'm gonna share a brief history of this pollinology. So the history of this pollinology is linked to the history of microscopy, with the earliest observation of the pollen made by an English botanist, Nehemiah Grew, in the 1640s. So after describing the pollen and the stamen, he concluded that the pollen is required for the reproduction of the flowering plants. So the Assyrians are said to have known the principles of pollination. They practiced hand pollination of date palms, but it is unclear if they recognize the nature of the pollen itself. So although this study has been very old, gone through prehistoric times, with the invention of compound microscope, the spore and pollen have been identified as morphological entity. So later on, with the invention of the scanning electron microscope and the transmission electron microscope, it have given a few dimension to the field of pollinology. So this pollinology is based on the Greek words palono, means to sprinkle, and pale or pale, it means dust. So in the pages of the Pollen Analysis Circular, one of the first journals of the pollen analysis. So others said that it was derived from the Greek word paleinin, means to scatter or to sprinkle, as many pollen grains or spore are easily dispersed or carried away. So let's go to the terminologies of the pollinology. So first is the pollen. So this term is introduced by the Swedish botanist Linnaeus in 1760, and it is derived from the Latin word pollinis, whose meaning is fine flower due to its granular nature. So pollen is haploid male reproductive body produced as a result of meiosis in the pollen mother cells containing male gametophytes of the angiosperm and gymnosperm. So in other words, it is an immature endosporic male gametophyte derived from the male spores or microspores in seed plants. So the spore, so this is an independent resting and dispersal phase of the independent gametophytic generation. So it is closely applied to the various reproduction body in cryptograms formed by sexual and asexual methods. So the spore is a cell which becomes free and direct development into new life. So it is the immediate product of meiosis of SMC, germinate and give rise to haploid gametophytic generations, which produces male and female sex organs. So in pteridophytes, they are isospores, microspores, and megaspores. So the pollen types. So these are the types of the pollen. So this describes the entire nature of a pollen grain, as specifically the size, shape, apertures, exene stratification, surface sculpture in a single phrase such as type A or Legionelliflorae type. So as you can see in the picture, these are the types of the pollen. So the dispersal unit. So this is the arrangement in which pollen grains are found at maturity and dispersal, also known as the pollen unit. So this is the types of the dispersal unit, the monad, the vesiculate or saccate, the dyad, tetrad, polyad, and pollinium. So the polarity and the symmetry. So they are determined with the reference to the development in the tetrad. Each pollen grain has a polar axis running outward from the center of the tetrad and the two poles where this axis meets the surface of the grain. So this is a proximal pole at the center of the tetrad and a distal pole at the outer surface. So the shape. So the basic three-dimensional shapes of pollen grains are often categorized as either globose or boat-shaped. Other shapes, such as thread-like, are restricted to a few species with reduced exenes. The first row of the picture shows a globose shape. And also there are also the boat shapes. So the apertures, so they comprise of modifications of the pollen wall, such as openings, thinnings, or thickenings of the exene or intine for the purpose of the interaction with the surrounding substrate, including the emergence of the pollen tubes during germination. So the exene, so this exene is actually part of the pollen grain. This is the outer covering of the pollen. So it is a protective covering. It protects male gametes from physical and chemical damage. It is made up of the protein sporopollenin. As you can see, it is thick. It is a thick layer. So the intine, so it is the inner, less hard layer of the pollen grain, made up of pectin and cellulose. So it plays a very important role in the maturation of the pollen grain and germination of the pollen tube. So pollinomorph. So this term is actually a microscopic resistant walled organic bodies found in the pollinological maceration. So this, as you can see in this picture, these are the pollinomorphs. Resistant, so this is the walls. So prepollen. So as Callner in 1970 stated that prepollen as any microscopic belonging to seed plant group, like angiosperm that germinate from the proximal side of the pollen. So thus, it is the functional pollen grains having trilete mark on the proximal side like spores and also germinate proximal. So trilete, so as you can see in this picture, the Lyginopteris prepollen has trilete mark where pawai shape siya, and this Dolerotica monolith prepollen. So monolith, so it has only one crest or layer that you or mark that you can notice in this one. So this is the comparison of the trilete marks. So the prepollen is the earliest pollen grains were not at all morphologically different from the spores. So in contrast, the modern pollen which have aperture on the distal side, that's why it has distal germination. So these prepollen are important intermediate evolutionary stage. So this transitional stage between the spore and the pollen is present in fossil spores of primitive Paleozoic gymnosperm. So prepollens are also bearing plants gradually became extinct during the Permian period. So the most recent known genus with prepollen is the conifer. So as you can see again in this image sa prepollen pala, the Doloretica monolith prepollen, so this is its type. So back again to the so the monolith prepollen, there is only a single narrow line. So this indicates that mother spore split in 4 along on a vertical axis. So in trilete prepollen, each spore shows 3 narrow lines radiating from the center pole, as you can see really. So also in this species, some monolith ferns were found to have larger distributional areas than trilete ones and to be more tolerant to a colder, drier climate. The paleopollenology. So this is concerned with the study of the fossil spores and pollen. So this branch is used to reconstruct the past vegetation and the paleoclimatic conditions. So this is branching sa palynology and other parts that I'm going to explain. So this is the first one. So in short, this paleopollenology is the study of fossil pollen grains and spores. So geopollenology, it is concerned with the spatial distribution of the pollinomorphs, their patterns of distribution on planet Earth, and the causes and effects of the spatial patterns and their impact to the ecosystem. So in other words, it is a study of the fossil pollen grains, like the pollens are used in finding age of the rocks. And Pollenotaxonomy is the morphology of spores and the pollen that have specific structure, including the structure and the ornamentation in the case of spores, and the number and position of the aperture and the ornamentation in the case of the pollen. So this is the constant spore and the pollen characters to the particular species is extremely helpful in the taxonomic identification of genus even at the species level. So the aeropollenology, so it is concerned with the study of the spores and the pollen present in the air. So the spore and the pollen present in the air show variation from one place to another place, like place to place variation, and one season to another season, like season to season variations. So this study of pollen found in the air and atmosphere that it also detects if the pollens present in the air cause different types of allergies like hay fever, asthma, and other cold, and these are called aero allergens. So the militopollenology is the study of the pollen grains present in honey type A and honey type B, and collected from the two different places. The variations in plants' flora may be observed after the morphological study of spores and pollen. And iatropollenology is concerned with study of pollen in criminology and medical aspects. Place or of crime can be judged by study of microspores and pollens collected from dirt under shoes, clothes, and nails. So this is related like example sa mga crime scene. It is helpful also in identifying this mga mga prints from the criminal. So also it is also the study of pollen grains and spores which causes allergy like eczema, asthma, allergic lung disease, mga bronchitis, hay fever, is susceptible also to humans. So this is related to forensic pollenology. So the copropollenology is concerned with the study of the pollen grains and spores present in external and waste products. So biogenic pollenology or pollen biology includes pollen stigma interaction, pollen viability, germination, pollen storage, production of haploid plants, and also in the plant breeding programs, which may be utilized in hybridization. So that's all that terminology is involved in the pollenology. So let's go to the pollenology and systematics. So first one is, we'll talk about the pollen grain formation. So it is formed from a pollen mother cell in a way which is almost unique in cell formation. So the nucleus nearly always goes through two divisions in the rapid succession, followed by two successive or simultaneous divisions of the cell, forming 4 new cells, which by growth and further development become a pollen grain. So in this illustration, you can see, so this is how the pollen grain is formed. So from the microspore mother cell, which is diploid, it undergoes meiosis 1 and then it undergoes to meiosis 2, which then forms into tetrad. Tetrad, which means 4 microspore cells, so it becomes haploid. Then it develops into the pollen grains, 4 pollen grains, and via mitosis, so this will have 2 nucleus and pollen tubes nucleus. Common arrangement observed, so the tetrahedral, and the tetragonal or flat arrangement. So tetrahedral is as you can see this picture, the grains lie in compact cluster and occupy the relative positions of the angles of a tetrahedron. And sa tetragonal or flat arrangement, in which they occupy positions of the 4 angles or a square or rhombus. So as you can see in this picture. So at maturity, they are generally quite separate and it is difficult to see just what effects of their contacts with each other were. So in few cases, however, the grains never separate but nag-remain sila united sa tetrads throughout their life. And others also, the occasional monstrosities occur. The grain remaining united at maturity is in the same relative positions in which they were formed. So a sort of like Siamese quadruplet. So we learn from study of this that the pollen grains may be associated with each other in all possible arrangement of 4 cells in contact, but the these arrangements most commonly observed, kanila lang silang most commonly observed na mga arrangements. So in pollen grain formation, there are 3 basic forms of pollen grains, like number 1, formed with the triradiate crest. So this is common among the Filicales and the primitive gymnosperms, but among angiosperms, this is known to occur only in Magnoliaceae. And I don't have any illustration of this because it is really hard to find, since na most of this is extinct. Most of this is extinct. So but I'm going to explain to you how these pollen grains appear. Each of the radii of the crest is a double ridge which may easily split open, providing lines of the dehiscence through which the sporoplast emerges at the time of the germination. So this form is actually common, to the Filicales gymnosperms. So the second one is the one-furrowed or the monoculpate form. So this form characterizes the grains of most gymnosperms, monocotyledons, and lower dicotyledons. So it may be elongate or rounded, but is always provided with a single germinal furrow, which forms on the side of the grain remote from the contact in the tetrad. So this furrow serves as a place of emergence for the pollen tube at the time of germination. So the third one is the three-furrowed or the triculpate form. So this type of grain characterizes all higher dicotyledons, though it is more often not profoundly modified, so that it is not easily recognizable. So this is the illustration of the three-furrowed or triculpate form. So in comparison with the monoculpate form, so it has 3 germ pores. So these 3 basic forms represent 3 different modes of response to contact stimuli received from the tetrad. So kani sila. So first, kay stimulus results sa flattening of each phases of the contact with the formation ridges between them, and then the second form, the one furrowed, kay no visible effect is produced at the sites of this contact, but their stimulus forces the formation of the pore or furrow at the opposite side of the grain. And this third one, the three-furrowed or triculpate form, the stimulus induces from the formation of the pore or furrow on each of the contact faces. Yet, these differences modes of response denote huge genetic differences, yet they are entirely unrelated. In Paleozoic, representative of spores and pollen, as you can see in this image. So these are some of the pollen illustrations from this primitive era. So if we examine the most primitive form of this pollen grain, we find that all of these are simple, rounded protoplasmic body enclosed by a coat in which an inner and outer layer can be distinguished. So the outer film, the protective, the inner delicate but impervious. The 2 layers are thus compatible to the casing and inner tube of the automobile tire. So this description would suit a fern spore equally well, which most primitive pollen grains very closely resembled. So kani sila nga mga grains kay they are kind of found among the fossils of the Psychodophilicales, a pollen grain of the Paleozoic Chrysotica, and is virtually the same as the fern spore gid sila. So it has a tri-radiate crest on one side, marking of the 3 phases. Marking of the 3 phases which were in contact with its 3 neighbors of the tetrad. Its opposite side, which was outward in the tetrad and which for convenience we'll call the ventral, was rounded. So So such grain had no germ pore or germinal furrow. So this is now the evolution of the pollen grains among the Chorditales. So still I don't have illustration for this since Chorditales is an extinct order na siya. Extinct order sa mga plants. So their pollen grains differed from those of Cycadophylicales in having a well-defined preformed furrow on the ventral side. So it appears that as the prothallus of this developed upon reaching the pollen chamber of the seed they were to fertilize, the invaginated furrow became evaginated and finally separated from the rest of the exine, which is the outer layer. Dayon, opening like a lid and then permitted to escape so into the pollen chamber of the motile spermatozoids. So these are fossil trees which lived in the Mesozoic period and were probably the precursors of our modern conifers. So Benititales, only the whole grain was partitioned in a few large cells, but more often only a few rounded cells were cut off and then pressed against the inside of the pollen. So this appears that ang prothallus nila kay developed upon reaching the pollen chamber of the seed they were to fertilize. So in the Araucariaceae, it is regarded by many as the most primitive of gymnosperm, exhibit the simplest way. So the floor of the furrow appears to have been evaginated and its exine thickened. So in this illustration sa agathis pollen, so the grains, as you can see here, the grains are spherical with the exine and uniform thickness throughout, so that one can not guess that they are bore any relation to one foreign grades. So but in others, also for the Araucaria clinky pollen, it's also a rim-like thickening is as you can see here, a rim-like thickening structure is evident, and which can only represent the rim of all but vanished furrow. This is in Australia. This species is in Australia. So in the evolution of the pollen grains among the Podocarpaceae, it is also primitive and similar to Araucariaceae at some way. So it's disposed of the furrow by developing a wing-like bladder around it. So this is so arranged that when the grain dries and contracts, the furrow dips, it causing the frill to buckle together, tightly closing the gap. So this ability to develop in this bladder frill was undoubtedly inherited from the remote past. So similar kay nakita nila sa grain sa ancestral Cycadophylicales such as Stephamus firmum caryodis. So kani nga kuan kay ang frill niya was developed probably as a floating organ for its possessor lacked a germinal furrow. Then it was a large grain, however about 100 microns in diameter, so that some floating device was necessary to enable it to reach its goal. So the example for this is this Podocarpus pollen. So, in this ang Magnolia Cae nga pollen kay simple ra siya and unmodified single furrow. As you can observe, there is only a single furrow. And also, this suggests that they are certainly among the lowest angiosperms and perhaps on a part where the Saururaceae and the Piperaceae, though not necessarily closely related to them because their grains are very much larger. So, in the Dremis pollen, it's somewhat similar sa ang kaning Magnoliaceae kay somewhat similar sa Dremis nga pollen and also to the Illiceae, but their particular interest lies in the fact that they remain united in the tetrad at maturity. So, since kaning tetrads kay tetrahedral, the grains are rounded or somewhat triangular and each has a single furrow facing outward and which is rounded instead of elongate, but it functions in the same way as it in the grains of the Magnoliaceae, bulging outward when moistened and dipping inward when dried, and serving as a place of emergence for the pollen tube at the time of the germination. So the main points of this interest in this grain lie in the fact that it shows plainly that the position of the single furrow is on the distal side. Is the distal side of the grain of the Magnoliaceae in relation to its tetrad. So as it is in the grains of the primitive gymnosperm. This relation is practically universal among the pollen grains and was first observed by Fischer and therefore designated sa Fischer na law. So, sa Illicium floridanum pollen, kay it belongs to the same tribe sa Dremis, pero quite different ni sila. It does not have any trace of the large single furrow. Instead, it does not have any instead it has 3 slender nga furrows which reach from the pole to pole. As you can observe here, there are 3 furrow, and also it divides the exine of the grain like into 3 equal lunes. So into 3 equal segments or lines. So these 3 furrows appear not to be homologous with the 3 furrows of the ordinary dicotyledonous grain. They do not participate in the harmomegaty, which like these grains are thin-walled and shrink by simple collapse in one of more their lines, and they do not provide places of emergence of the pollen tubes. Let's go to these interfamilial or higher level groupings with the angiosperms in terms of their maximum parsimony optimization. So here is this clade of this So this is the summary of the evolution of the pollen characters sa angiosperm. So ang colors and the patterns kay na indicate sa characters nila like in this legend. So color red is the symmetry, this blue is the basic shape, the polar outline ang black, and then so on. So kaning illustration kay naay 21 ang kanang character state changes inferred by this empty optimization of the pollen and then under this species naay polymorphic data points pud na missing. So in this figure, this 21 character state changes is defined interfamilial or higher level groupings sa angiosperm. So kaning 45 nodes nila kay kaning nasa phylogeny kay naay 15 is associated sa kaning one inferred character state change niya. Then naay 4 associated with 2 changes and naay 1 with 3 changes. So the node associated with the greatest number of inferred character state changes is that linking of Zingiberalis and Poales. As you can observe here, naglink ang musa. As you can see here, naglink ang musa to this Poales. So here. So as you can see again in the phylogeny kay kaning Zingiberalis and the Poales kay share transitions from bilateral to radial symmetry, which is the character 3 color red, as you can see here. The Musa and the Taifa. Taifa is the Poales. So, also, the both shape to globus pollen pod sa character 4 and the kaning elliptic to circular pollen nila kay in polar outline. Kaning color black. So, 3 characters which are not of course independent. So, the 4 nodes marked by the 2 inferred character state changes are the root of the angiosperm itself, defined by a gain in the suprathecal elements. So, the transition anin lang elevate to the columellate infracticum structure, and the root of the monocot switch transitions from globose to both shape, and the circular to elliptic pollen anin nila. So, here are the structure. So, and then further reverse line within the clade. So, ang Zingiberalis and the Poales nila. So, ang root pod sa kaning comilinids, kaning Aricales, Cominiales, Poales, and Zingiberalis, ang characterized nga transitions sa ila kay from granular to smooth aperture membrane and perforate to reticulate tectum sculpture, and then further modified several times on terminal branches within this clade, and the asterids plus the Caryophyllales represented by the spinacia defined by a further gain in the suprathecal elements, and the transition po sa reticulate to perforate tectum sculpture. So these key nodes characterized by a single inferred character state changes under the MP, and this includes the linking of all the angiosperm except the Amborella and the Nymphalis, like they have polar outline elliptic to circular, and that linking all angiosperm except the Anita grade. So, the root of these eudocots also and the root of the asterids are simple to compound. Napod sila simple to compound na structure. So, let's go to the palynological technique. So, 3 major instruments sa palynological technique kay light microscopes, scanning electron microscope, and the transmission electron microscope. So, we're gonna term the scanning electron microscope as SEM and the transmission electron microscope as TEM. So, light microscope, it is the original and still basic method of studying pollen grains. So, kaning pollen being since kaning pollen being decisive is ang palynology is totally dependent upon the use of various types of microscopes. Good. So, sa step sa light microscope, first, kay ang palynological material is normally first subjected to an acid treatment or acetylysis. So, dayon, ang acetylyzed pollen grains are mounted on a permanent slides. Dayon, why use that treatment acetylysis? So, it functions as, it is the removal of the pollen protoplasm, a part of the pollen wall, and a part of the pollen wall since the intine material that may cover the pollen surface, such as oil droplets and protein, pollen kit, and trypsin should be removed. So then it causes the outer pollen wall layer, ang exine, to turn from its normal light yellowish green to a darker amber color. So both of these changes kay nagresult ni sila sa production sa greater pollen contrast. So, and it permit also the more detailed study of pollen grains than is with possible of the non-acetylized material. So here is the example. So as you can see, muna siya ang result sa light microscope. You can really see na visible gyud ang kaning part sa microscope, like ang part sa pollen, like ang kaning intine og exine. but not in really like very much closer. However, kaning si Walker kay yang gi-modify ang kay Earthman nga standard acetylysis method. Na siya gi-modify na method like ang kaning na siya duha ka features nga nahunahunaan. The introduction of a less drastic means of preparing pollen than boiling, which frequently causes a number of grains to be damaged. And number 2 is the development of the method of acetylysis that can handle a large number of individual pollen samples rapidly. So, iyang modified acetylysis kay ang pollen is placed in cork centrifuge tubes with standard acetylysis fluid, 9 parts acetic anhydride to 1 part concentrated sulfuric acid in a oven overnight at 50 to 60 degrees Celsius. And the material is then washed with once lang with glacial acetic acid and 3 times with water. So, ang number 3 kay acetylized pollen is then mounted in glycerin jelly and slides ringed with an air-drying phenolic varnish in order to make them permanent. Taxa with reduced exin such as Lauraceae, Musaceae, etc. and are subjected to KOH treatment instead of acetylysis. So, this is the use of the disposable, as you can see here, the disposable pasture pipettes was found extremely helpful in transferring a mixture of pollen and glycerin jelly evenly to slides, especially when the available material is scanty. So, the fourth step is the cork centrifuge tube containing pollen material and varying concentration of the KOH up to 1N is then placed in the oven overnight at 50 to 60 degrees Celsius. And the step 5 is the material is then washed to 3 to 4 times, stained in basic fuchsin to toluidine blue and or Alshan blue and mounted in the glycerin jelly. So kani siya nga step kay when pollen mounts prepared by these techniques may be studied and photographed in the light microscope at the level of the whole grain topography to determine the aperture type of the, for example, then at highest magnification using various focal planes to analyze exine structure and sculpturing. So one may also photograph optical sections of exine, especially as seen in median optical view. Thin sections of the exine may also be prepared for study in the light microscope using paraffin-embedded material and an ordinary rotary microtome. So, in scanning electron microscope, this technique greatly complements light microscope studies because of many advantages. So, iyang advantage kay, these are the several advantages of this scanning electron microscope. So first is great range of magnification, relative ease with which specimens can be prepared for study, considerable depth of focus. As you can see in this image, very kuanjid gayo kanang klaro ang kaning parts sa pollen, and ability to show the pollen surface good independent of underlying structural components. And also, it often aids in the interpretation of the confusing or ambiguous LO analysis seen in the light microscope. Mas way better din siya kaysa sa light microscope. So, also, one may deliberately break pollen grains and study them with this microscope in order to acquire a three-dimensional view of the internal wall structures such as this. So, ang steps for scanning electron microscope kay number one is the pollen samples should be subjected initially to acetylysis treatment, then transferred from water to double adhesive tape and allowed to dry. Samples may be carbon coated before coating with a heavy metal such as gold palladium prior to their placement within the microscope. So in the transmission electron microscope, it is vital for high-resolution micrographs necessary for complete understanding of the pollen wall. The main advantage offered in this technique, besides higher magnification than the light microscope, is the ability to delimit and resolve ecdexine and indexine and demonstrate the presence or absence of a foot layer. So also, these measurements of the different wall layers may be carried out much more accurately with the transmission electron micrographs in the case of the small grains with thin exins, and some cases both cross and tangential sections are necessary to elucidate the nature of the pollen wall. So these are the techniques na sa palynology. So thank you for listening.